Wonderful. It is my pleasure to introduce today's speaker, Montana Smith. Montana is a data scientist at the Pacific Northwest National Lab. She started working in the lab researching soil microbial ecology and over time found a passion for data reusability. Yes, standardization and sharing. Now she uses her skills and knowledge for working in the field to help build the bridge between experimental and computational scientists. Her goal is to ensure that the tools and knowledge exist for data generators to capture data in a way that is findable, accessible, interoperable, and reusable. So I think this is fantastic. She's clearly kindred spirit. And today she's going to be presenting on the NMDC, Advancing Microbiome Science Through Fair and Standardized Metadata and Data. And with that, I'm going to shut up and let you go.
Thanks so much. All right. So I'm just gonna introduce the National Microbiome Data Collaborative, or NMDC, and what we're doing to, as Lenore said, try to bridge the gap between experimental scientists and data scientists to enable fair data and data reuse. So I will share my screen and hopefully grab the correct one right off the bat. There we go. Okay, you should see some slides. Okay. So I'm at Pacific Northwest National Lab. I'm at the Environmental Molecular Sciences Laboratory, which is the DOE user facility for DOE, and my role within the NMDC is both as a connection for our users, but also for non-DOE data generators and anybody who cares about fair data and data standardization. So I want to start off to just get a kind of a sense of the room. So if everybody could use the raise hand feature on Zoom, and tell me if you know what the NMDC is. Anybody? All right, got a couple. Okay. So next, if you can put your hands down and then tell me if your lab or your research area collects standardized metadata. All right, Clay gets a gold star. Okay, next, using the same hand feature, have you ever tried to reuse someone else's data? Who's ever tried to reuse somebody's data? Yep, a few hands. Okay, a good number more. Cool. Okay, now thumbs up or thumbs down. How was that experience for you? How easy was it to just find somebody else's data and start running some analyses up or down? How was it? That's a good one. I like that. That's a first. Yep, thumbs down. Right. Yep, that's pretty on par with what I expected. Yeah, so data reuse is really hard. And unfortunately, we're still struggling with this after a good amount of time of trying to make it better. And it's still an area to start improving. So some of this difficulty, right, comes from the fact that science is always advancing. We're constantly discovering new and better ways to interrogate our samples, generate or analyze data, and this is ever growing, and which is great. But bigger, it's always good to get bigger, better science. But the scale at which data, and especially omics, multi-omics data, has been generated currently really outpaces our data, especially if you want to be able to share data. And because of this outpaced development, accessing data sets for reuse is a really complex process. Every lab has their own system or for their capturing and tracking their own samples or source, and that doesn't scale well across labs. And sometimes not even within your own lab can you necessarily track data. As I said, I'm at a DOE user facility, and so basically what that means is, if anybody's not familiar, you write a proposal. If you get funded, you send us your samples. We run those samples on our fancy instruments. We generate data. We give that data back to you. But there's a big process there of actually generating that data that things might get confusing, or you might decide that you want to rename your samples, or something might change. And now you have to figure out how to map that data when it comes back to you. And so even within your own lab, it can be hard, let alone actually trying to interoperate and connect across labs. And so we end up with this science spaghetti figure that we're seeing here for trying to figure out how data relates. And this is what the NMDC is trying to address and really get at. We aim to streamline this process of discovery and access so it's easy and clear to know how any source sample or data goes from source sample to data and to a targeted result. So the National Microbiome Data Collaborative is a data discovery platform promoting open science across broad and diverse communities. As I said, we are a DOE-funded program that is a collaboration across 3 national labs. We have members at Los Alamos National Lab, Berkeley Lab National Lab, and Pacific Northwest National Lab, where I'm at. And then the 2 user facilities are Environmental Molecular Sciences Laboratory, or EMSL, and the JGI, which is the Joint Genome Institute. The NMDC's vision is for connected data, people, and ideas to advance microbiome innovation and discovery. And our mission is to support a fair microbiome data sharing network through infrastructure, data standards, and community building that addresses pressing challenges in environmental science. And at the heart of this, and where my bias comes in, is that that heart is really the fair and connected data is metadata. To really be able to link samples and link data, you need good and consistent metadata. And accomplishing this really means that you have to go also beyond the current state of data, right? So kind of like this puzzle analogy, right now we kind of have these distributed data types that it's helpful maybe for your specific section or area of science or piece of science. And then we can get maybe into it like an aggregated data where you can maybe group some data, but we're missing the integrated piece, the big piece that shows how to get the full picture of microbiome science. And to get to this vision of integrated data and well-captured and open data, open and accessible data that is part of FAIR data principles, we really have to get to focusing on what it means to accomplish this, right? How do we really get there? So FAIR data being findable, accessible, interoperable, and reusable, and open and accessible data that enables for expanded science to advance and to pair your data with other data to lead new discoveries. So this open science and open data goal that enables data sharing and expanded research questions within the NMDC, we support projects in microbiome science that in a standardized way so that you can combine study questions. So for instance, if we have these 3 projects that are currently represented on the NMDC data portal, they all had their distinct questions, right? Focused around carbon, focused around microbes in watersheds for nitrogen, focused around biogeochemical cycling within deep subsurface environments. But they all had their own research questions. They generated a lot of really great data. But what might be a good connection? How might we be able to link these studies, or what other results can we find from this data? And we want to enable researchers to do that as, again, data reusers or to complement their own data and really, again, just expand this question within science. However, data sharing and management requires a really methodical approach and a lot of thoughtfulness be put into your data lifecycle. Where you capture and how you capture metadata and processes about your experiment or your samples or your process has to be done really at every step because metadata comes in everywhere. There's all types of metadata, which I'll get to. So during your experimental design, it's vital to plan for every step and be intentional about that process for collecting, storing, processing, pre-texting the data for your search. So you can kind of think of this data lifecycle where you have your own research question, you come in, you create, data in a fair way, you run a process to generate your data, you analyze that data, you want to preserve that data, but then you need to provide it so that other people can access that data, right? That's part of FAIR, that's part of being open and data and having data sharing. And when you do that, somebody has the ability to reuse and generate another research question and go about this cycle over and over again. Again, just really expanding the power of your data. And too often, the pieces here are an afterthought, right? Somebody's already generated hundreds of samples, they've already generated hundreds of data files, and now they want to share that data post-publication, and you have to kind of go backwards and figure out how to capture that metadata and information, and very high quality, valuable detail gets lost. But we also can't just make up our own capture for this information, right? So let's say that you do a very good job of capturing information about your experiment, your metadata, your study, your samples, but you do so in a way that is tailored for your lab, right? Well, now you've kind of lost that interoperability piece. And this is really part of what makes data reuse hard. The royal we, because I don't think I was part of this, did an evaluation of SRA data to just get a sense of like, what does the metadata look like? And the results were not great. There was quite a big gap in the metadata standards and data management standards. And it was really obvious that this lack of completeness and compiled metadata would result in a lack of ability to compare across studies. So NMDC is working to shorten this gap in standards and improve fair data. So that was probably like 10 sides to all just say the same thing, which is data is very important. We should be open and fair, and we need standards and standardized metadata, which I'm sure everybody here probably already knows and cares about. So now I'm going to kind of walk through some of the details around NMDC and give some information about NMDC and what we're doing to kind of try to fill this standards gap and this data sharing gap with our 3 products and the tooling that's centered around these products to provide fair and open data. So I'm going to start with the NMDC submission portal and field notes app, which you'll hear a lot about from me because I am the submission portal product owner and, again, totally biased around metadata. So as I said, to accomplish data sharing and interoperability, clear and concise metadata is required and standardized machine readable formats. And to help research understand metadata, the when and where it applies, we at NMDC think about metadata within these 4 categories. So we have sample preparation, data processing, and feature metadata. Sample metadata is what defines your experimental sample and the environment from which it was collected. Preparation metadata describes how you go from that environmental sample and its environment to a sample that has been processed and is like ready to be run on an instrument. So how did you go from soil to extracted DNA? How did you go from sediment or water to digested peptides or derivatized metabolites? And this data processing and the data processing and feature metadata are how you process your data. So what quality filters did you put on? What reference databases did you use? And then feature being what results did you find and what were the conclusions you came to? So the NMDC submission portal right now captures the sample metadata piece. So again, that environment and the starting material, the experimental material that you start with. It's the when, where, what, how of your sample and the experiments that you've run. And it's the information that one would need in order to understand the results of your samples. So let's say that you go out into the field and you collect soil. I'm going to keep using soil as my example because soil ecology is my background. Sorry. But let's say you go out into the field and you collect soil and you collect it from 3 different locations with 2 different cropping types and irrigation versus no irrigation. Within any minimal metadata standards, irrigation, yes or no, probably won't be required because not everybody's going to have irrigation, and that doesn't matter for every sample type. But it's about identifying those pieces of metadata that are important for your experiment. If I see a separation in data and I can't explain it from the metadata, it means, well, maybe I missed that irrigation metadata piece. So that's kind of what we're trying to capture in the submission portal. And as I said earlier, just collecting isn't necessarily the start and stop of it all. It does have to be done in a standardized and machine readable way, even if it's readable to the human, doesn't mean it's readable to the computer, right? So for this example here, if we have one scientist who calls their material water and groundwater, another one who says aqueous and ground liquid. I don't know who's saying ground liquid, but maybe they are. You lose the ability to compare within a machine, right? Again, as a human, you can probably tell, but a computer won't know. And if you search for one, you won't get the other. So this lack of validation and consistent and standardized metadata means that the data will look incomplete and incomparable. But when you standardize it and being consistent within your formats and your terminology and your units, now you as a human can compare, but so can the computer. We can run a query on a database and pull out this information without necessarily having to worry that we have to string together a bunch of types of queries or know what other values might be there. So collecting data and metadata in a standardized way that's machine readable and interoperable does, again, require a standard. And those standards have to satisfy multiple data types, multiple metadata types, multiple study types with lots of different data sets and data types. And unfortunately, we've got every single community trying to come up with a standard where everyone has figured out something that works for their community. So that's an improvement to individual labs, but we still have this problem of being able to compare across community standards. And we end up with this, I don't know if anybody else is familiar with XKCD, but their comics are very entertaining. And we end up with this kind of situation where everybody tries to make a standard that will work for everyone, and we just end up with another competing standard. So for NMDC, our approach has been to not make any standards. We do not have an NMDC standard. Rather, we are working with communities to improve the existing standards and provide connections and linking and association. So our base schema is structured around the MixS standard, and then we have attributes within our schema to allow us to link out to other standards that exist. So because we're leveraging existing standards, and I said MixS, so our base schema, and we use the standards from the Genomic Standards Consortium using their minimal information about X or any sequence. So MixS, GSC, they have environmental extensions, which provide a community-built metadata standard for structuring metadata and required information. If you've ever submitted to INSDC, any of the INSDC members, then you've probably seen these metadata extensions. So that we use the GSC. We work very closely with the GSC to ensure a two-way communication for improvement and expansion, and that's what we built our schema off of. We also include and have implemented the Genomes Online Database, or GOLD, from the JGI for environments. This is, again, another metadata data storage that the JGI uses. And so we provide that information within the NMDC. And we also are leveraging existing ontologies like the ENVO ontology to drive standards to improve community experience. And all of these standards and all of this metadata is captured via our submission portal, which provides a place to complete, as I said, sample metadata in a standardized, structured way that is schema compliant and valid. And so just kind of stepping through, we usually give almost a full hour workshop on the submission portal, but I can't spend all of the time talking about my favorite piece of NMDC. So I'll just give you a quick overview of what this submission portal can do. So the submission portal is built off of a data harmonizer framework, and it has kind of like a hands-on table in-sheet view from a UI. So this is actually on the web browser in the UI. But if you hate working in this, you do have the option to download the Excel template. You can fill out your metadata in Excel, so you can retain all of the power of Excel, like vertical lookup and concatenate and other tools that people use to generate and capture their metadata. And then you can import that Excel file into this web application to then actually use all of the tools. So once you've imported your metadata, we have in-sheet real-time validation. So this runs a metadata and compares it to the schema, and it lets you know if any of your metadata is invalid or missing, and that is using these color codes down here. So if a field is yellow, that means that it's required, and if you get a field that pops up this darker pink, that means that you've left a required field empty, or the lighter pink means that you've got some formatting error. So we found that this really helps people maintain that activation energy for metadata, right? You don't have to hit submit and then wait to find out if your metadata looks good. It's this real-time validation, so you can really crank through it in real time. We provide guided enumerations for terms that have specific structure. So these dropdowns are interactive and provide all of the terms that are valid, so you don't have to remember if it's metagenomics or metagenome and then end up with something invalid. Again, it's real-time validation, and we provide the enumeration right there on hand. We have this column help feature for every single term that's within this like spreadsheet interview or spreadsheet view, where you get the column description, and also in this scrolly thing is an example and structured guidance so that you can understand what you're looking at and what a slot is for, as opposed to needing to jump back and forth between sheets. And similar to Excel, we have a find and replace tool. So if you, do write metagenome and you meant to write metagenomics, you don't have to change every single field individually. You can use the find and replace tool just like you could in Excel. And then lastly, up here, we have this jump to column and a show hide feature. So if you've ever submitted metadata using the MixS formats, you probably know that there are a lot of columns, there are a lot of terms, and it can be overwhelming and hard to sort through. And so using this tool, you can filter to just show required fields, just recommended fields, or you can say, if you know that you want to fill out pH, you can start typing the word pH and it will jump you to that column. So this is one of the tools that we've created to try and lower those barriers for metadata management, metadata standardized collection for researchers. And then next I will also, I was supposed to take that one out. I'm going to just go to the one that actually has the title. And then I'll also mention we have the NMDC Field Notes app, which has just launched for beta testing. So this app is on your phone. You create a study, and then you can take that out into the field and you can collect metadata in the field in real time while you're doing your field work. It uses some auto completion and connection information. So it can pull your latitude and longitude. It can pull date and time on your phone to auto populate some of these metadata fields for you. And just kind of give you the opportunity to be able to capture your standardized metadata in real time instead of having to, write it on a piece of paper and then come back into the lab and record it again. And it connects automatically directly to the submission portal. So you don't have to capture all of your metadata when you're in the field. You can capture what is field relevant. And then when you come back into the lab or into your office, you can then access this on your desktop. So as I said, this app is available and in beta testing. So if anybody's interested in being a beta tester and would like to check it out, please let me know and I can get you connected. All right, so moving on, I'm going to jump into NMDC Edge, which provides a place for streamlined multi-omics data processing. So NMDC supports multi-omics data, so metagenomics, metatranscriptomics, metaproteomics, and metabolomics. We're currently in the process of getting a lipidomics workflow in, and these data types are rapidly becoming the most effective way to interrogate the microbiome. But as I said at the start of this talk, the infrastructure is struggling to keep up with the data generation. And also struggling to keep up in the compute power that's needed, right? And adhering to fair data and interoperability, right? It's all like difficult to manage within even just your own lab, let alone across labs for data sharing. Additionally, how these data are processed can be really variable, which lacks in cross-study comparison and data reuse. If somebody uses a different version of a pipeline or a different method of identifying peaks or taxa, you may not necessarily have the ability to compare. Or if you do, you're going to end up with some artifacts or results that aren't necessarily because of the research, but because of the data processing methods. So NMDC has integrated state-of-the-art open source bioinformatics tools into standardized workflows for processing raw multiomics data to produce interoperable and reusable annotated data products. So standardized workflow is available for all data types that are hosted within the NMDC, and these workflows are containerized and available on Docker, so you can access them there. And importantly, NMDC has recently expanded its schema to allow for metadata-informed workflows for mass spec data. So I don't know if anybody here is familiar with mass spectrometry data, so metabolomics and metaproteomics and lipidomics, but there's a lot of metadata pieces that can influence the way that you analyze the data. And so we are in the process of rolling out some schema expansions to allow for that metadata to inform the workflow. And if there is any change within the workflow across the studies, then you can at least identify where that metadata is and be able to evaluate that. So the NMDC workflows, while containerized and available on Docker, as I said, this is our other product. So NMDC Edge provides a user-friendly UI for workflow execution. And importantly, especially for metagenomics data, that workflow execution compute power is provided by NMDC. So if you have your own multiomics data and maybe you want to compare it to NMDC data, or you just want to be able to use the NMDC workflow, but you don't want to deal with a Docker container, you can come into the NMDC Edge and run your data here, and it will be processed in the same way that all of NMDC data is. Okay, next we have the NMDC data portal and our API. So as I said, NMDC provides standardized workflows for multiomics data. These workflows are used to process data and workflow outputs, and those workflow outputs, so the process data, can be found on the NMDC data portal. I want to emphasize here that NMDC is not a repository. We don't store raw data. Rather, we provide a platform for data discovery and standardization for data integration. So Amazon for your data, right? That Amazon's got lots of vendors and people who share their products, and then you come to Amazon to find those products, and that's kind of like NMDC. So in the data portal, you can search for studies, samples, and data by the standardized metadata that we highlighted earlier that is collected via the submission portal. So things like geographic location, function, and many more of these metadata fields are available for search within the data portal. And because NMDC provides these standardized workflows, again, when you discover data, you will know that the results are interoperable because they've been processed the same way. Okay, so another quick show of hands / check-in. Who knows what an API is? Anybody? Okay, so most people, so I don't have to explain an API too much. Okay, so an API just basically provides a place to request data to serve up information. So you can kind of think of an API as your waiter at a restaurant. If you're sitting at your computer and you are wanting to access some data that exists somewhere, but you don't necessarily want to tap directly into the database, you can send a request to an API, a query request. That API then will query the database for you and serve that data back to you in the designated format. So the NMDC has a public API for data and metadata access. So no matter whether you're more comfortable with using that UI or the API queries, NMDC data is open and available and accessible. And one example of use cases for the NMDC API can be found with some provided Jupyter notebooks that we have. So kind of as a tool just to start to show data users how they might access NMDC data, ways to explore the data and analyze NMDC data using this standardized metadata, we provided these Jupyter notebooks. So if you're familiar with Jupyter and you think NMDC may be of interest to you, I encourage you to go check them out. They're open and provide, as I said, examples for how you can access NMDC data. Okay, the final piece and the last thing that I'm gonna to highlight is for our community engagement program. So NMDC is following a user-centered design. So basically what that means is we want to build tools for the people, built by the people and with the people, the community's input. So following this user-centered design process means that all of our products are created based on user feedback and conducting of user interviews and user research, so that again the user can give us so that the user can give us feedback on what they would like to see. And we want your feedback. So as a if you're interested in participating in user research and providing feedback to the NMDC, please visit our user research site for more information. And I'll just kind of step through a couple of the programs that we have that are how we've done some of this, how we've received some of this feedback. So as part of our, we have our NMDC Ambassador Program and our Champions Program. So this first program, the Ambassador Program, consists of early career researchers in the microbiome community that are committed to making microbiome data fair. NMDC ambassadors spread awareness of metadata standards, data stewardship, and data standardization within their communities. This is a one-year program with a really focused contribution and focused on development of data sharing and interoperability. So our ambassador program brings in a yearly cohort. They participate in a variety of data standardization and data stewardship trainings that are led by NMDC. So kind of like a training the trainers program. And then they are required to host 2 events for their community in partnership with NMDC to really, again, kind of try to spread this awareness of data standardization and fair data practice. Our second program is the NMDC Champions Program. So this program, unlike the Ambassador Program, is open to anyone. There's no time limit. It's not just early career, and anybody can join. Our champions gain early access to NMDC testing and provide, again, a lot of this valuable feedback on our development and our program goals. And then in addition to the 2 targeted programs, we also just in general have a lot of open communication and engagements and collaborations with many other groups, teams, consortia, and community to expand data integration and interoperability for fair and open program. For fair and open data within our program. And because data sharing requires a community and a community working together is the only way that we're going to avoid, again, these 2 less than ideal situations within data sharing, right? Proliferating extensive standards and sending somebody a Word doc with an embedded photo is not necessarily the best way to share your data. We want to get standardized structures, standardized databases so that it's open and available to everyone. So if you would like to get connected to our community, you can reach out to us on any of our communication channels. We are on Slack, X, Instagram, and LinkedIn. You can send an email, and then you can also go to microbiomedata.org to sign up for the NMDC newsletter. And with that, I would like to thank my team, our partners, and our funders for funding us and allowing us to do this work. And then also thank you to the Ag Bio Data Consortia for having me. And I would love to answer any questions you have about the NMDC.
Excellent talk. Thank you so much for that. We already have one question in the chat from Tanya. the submission portal looks very user-friendly. A bunch of questions about it to follow. What kind
Great.
of usage do you see for it per month? Like, for example, can you tell how many of the features are being used? Do you get more submissions with more metadata with the new portal versus how you received data before?
All really great questions. Okay, so in order, what what kind of usage do you see for it, say, per month? On a So I can tell you that from a general just like testing and poking around and people experimenting with it, especially because we do highlight this in workshops pretty often. There's a lot. We have we do delete test submissions, like if it's obviously from a workshop pretty regularly, but there's still a couple hundred submissions on our submission portal. As for real submissions, we are connected with and collaborating with the DOE user facilities. They are encouraging users to use our portal to complete their metadata, so they only have to do it one time, right? As of right now, if you don't use NMDC, you should capture your own metadata in your lab for your own records, but also MZIL has its own structure, and then JGI has its own structure. So that's 3 different times that you have to do your metadata for yourself. NMDC, JGI, and MZIL all in one sweep, and then we manage the sharing out of that metadata. So if something is cared by both facilities, we send that out to them. You don't have to navigate that. And so we do a quarterly report, and this last quarter, I think we had I think we were at like 10 real submissions from users, and not all of them were DOE users. There were definitely some names in there that I didn't recognize. And so we also don't want to limit this to just DOE. We also don't necessarily want to limit this to just people who have data to share to NMDC right now. The submission portal is housed separately from the data portal data and what gets processed and shared publicly. So you can submit your metadata to the NMDC submission portal, and it will stay there and be private until you're ready to release it. Can you tell how many of the features are being used? I don't know. I have never looked into that. That's a really good question. We do some Google Analytics to like track other pieces of our products, right? Like where people are clicking and what they're doing. That's a really good suggestion to see if like all of these features are being used, and if not, what can we make room for new features? I think a Google Analytic would be really helpful for that. Do you get submissions with more metadata with this new portal versus how you received data before? Yes, so much more. And on top of getting more data, it's also structured correctly, which is, I think, actually the big benefit, right? So because we follow the MixS standard, we follow the minimal information requirements. So say, for instance, if you're submitting for soil, there's the standardized terms that are required for every sample, which is like sample name and the environmental triad. But then if you're collecting soil out in the field, we need to know the geographic location name, the latitude, longitude, elevation, and depth. I think those are the only required ones for soil. But we're no longer getting a bunch of junk in a lot of those fields, right? So depth is required to be reported in meters, and in specific structure. We implemented this because we did a evaluation, I think, of NCBI, and there was so much variation in the way that people reported depth. One of my favorite ones to highlight is somebody wrote centimeters with an S instead of centimeters. So it's like you can't do that anymore, right? There's better structure there. And so we are in general getting the minimal requirements every time. The added tooling seems to allow people to be able to, again, if you have a temperature treatment, find the temperature columns, and then just the general structure of the data.
So I want to ask a sort of a follow-up question, because we know, we build these things and people don't necessarily come. We as curators and database people totally understand the importance of all of this, but part of the issue seems to be incentivizing people to do this work. So one of my questions is, do you track, not necessarily who, but how many times data is accessed? So to be able to report back to the submitter, like, this is how much your data is being reused, and therefore they can get some sort of credit for that. And then, is there any relationship that you have with publishers, for example, about, requiring this information as part of publication, because we sort of feel like the publishers are an integral part of this ecosystem as far as really incentivizing participation.
Yeah, absolutely. No immediate publisher connections that I'm aware of, but we're a really big project, so I don't know the corner of every piece of that. There might be conversations that I'm not in. So I don't think, but I don't think so. So that initial question. As for attribution and credit, so within NMDC, when you make a submission or when you share data to us, either, relic data, old data or new data, we provide a place for you to attribute publications that are associated with your data. So even if you published and the data lives elsewhere and it wasn't published using NMDC workflows, you can still link all of that information. We provide credit roles for attribution and how people contributed to that study, again, within NMDC and how that's shared. We do have guidance for if you publish on shared NMDC data, how you should cite NMDC. And of course, there is definitely always you should also cite the original DOI for the data, right? And so we also share data DOI so that people can access that data, the actual raw data, right? So again, NMDC provides these standardized workflows. That workflow is great for data discovery and for data interoperability and being able to ask that initial question of like, is this the data I want? But maybe you want a different cutoff, right? Maybe you have a favorite tool that we don't use and you want to reprocess the data. That would mean that you're not necessarily using the exact NMDC data, and you should definitely still be referencing the researchers responsible for the original data generation.
Are there any other questions from folks? Raise hands or drop something in the chat.
Yeah, it looks like Monica.
Yeah, thanks, Montana. My question was, so you said that the NMDC is not a repository, but you collect a lot of metadata. So where does the metadata and data live once it goes through the NMDC workflow?
Yeah, so if we go through the whole process from start to finish, right? So the metadata is initially stored and captured via a Postgres database. That's on the back end and private, very only open to NMDC members, right? It's not public. When data is publicly available, / when the submitter says, hey, I'm ready to share my data, we have a we have a ETL script that pulls the data from the Postgres and stores it into our Mongo store. So we have a Mongo for all of our metadata. That is our central metadata store. And then we have the workflows have tooling to reach out and find the raw data. So part of the information that we ask that you provide is, where is your data housed, right? Can we find it at NCBI? Is it at EMSL? Is it on Pride? Where is the data archived, right? What is that data repository that you use? And then we query that repository to pull the data in. As of right now, right, if you've got a retrospective project or an older project where you didn't get this metadata completed ahead of time, there is still some data splunking that we have to do to make sure that we can map the metadata to the data, and that, if the sample names are consistent, and making sure that we can provide those correct associations. NMDC workflows get run. That process data then gets stored. I think it's still in the Mongo. It might actually be in another Postgres. I can't remember. But then from the Postgres, it gets shared on the database. So it's kind of a linked combination of databases.
Okay, yeah. So then the metadata that you collect, so say I'm submitting my soil metagenomic sample through NMDC, and once I go through that system, I also submit it to an INSDC database, as you were suggesting, because that's where the data repository is for that, the recommended one for that data. So the metadata that you all take so much care to collect and structure, can that also be submitted to NCBI so that is also the metadata repository, or how does piece works so that the metadata, as you said, you were not a repository, and so I'm assuming like there's the metadata itself is also not the authoritative repository for that. So how is there sort of a easy connection so that if I create my metadata there, I can also submit it to NCBI and they'll take all the fields?
Yeah, so NCBI uses the Access doesn't necessarily require that you use them. Also, it's just a spreadsheet with no validation, right? So that's why we end up with a lot of junk sometimes in the metadata that we find. And so the format is already there. So you can download the metadata once you've completed it on NMDC, and it should very easily upload to NCBI. We did also just pilot our first project of NMDC pushing data and metadata to NCBI for the submitter. So then kind of also cutting out an additional step, right? So I mentioned that we have these connections with these DOE user facilities so that if you want to share on NMDC and you're sending samples to JGI and you're sending samples to EMSL, you can do your metadata in one place. You don't have to do it in all three. Well, we are also now adding NCBI in there. So if you also plan to submit to NCBI, you can complete your metadata in our submission portal and we can push that up to NCBI for you. And we hope to expand that beyond NCBI for anybody who cares about mass spec data.
Yeah, that seems really great to do that. Are there any other questions for Montana? Yeah, no, one of the things I was sort of thinking about, with these forms, right? It's like the easier it is that you make for people to be able to submit the data, the much more likely they are to be able to use it. And one of the things that I really appreciate is the ability for people to sort of, be collecting the right kind of data at the beginning. And we've had some conversations about the idea that like you really need to be fair from the start. So it's interesting for me to think about working with facilities. So you are a facility, like, are there other facilities that do things like you do that you're sort of trying to onboard? Or are you like, we are it, this is where you go? Or, are there different institutions that you could partner with to really spread that around?
Yeah, great question. So all of NMDC is open and publicly available. So if you want to stand up your own instance of a submission portal, you can. We've collaborated with the bioenergy research centers. They are in the process of, I think, trying to improve their, data structure and data management, and they've started in looking at using our tooling. So we're not necessarily, the one ring to rule them all. You don't have to use only our system. We do want to collaborate and reach out and connect. And allow people to use our tooling if they don't have anything. And we actually, so this data harmonizer framework is created by the Show Lab in collaboration with Damien Dooley, who's in Canada. So this tool was actually created for COVID metadata collection. And so it was also that collaboration actually where we found this tool. So yes, definitely want to make those connections and share. And again, that slide I showed with all the communities that we work with.
Tanner?
Uh, yeah. Can you hear me?
Yep.
All right, good. Because I'm using this phones for the first time with this. So yeah, I think it's great work. What I'm thinking of is that maybe following up on Leonor's question is about the collaborations. I think there are so many DOE labs that their collaboration is so natural, comes to them naturally, and I see that you're also collaborating with NASA as well, and Pankaj was involved with the standards, which is great. But I'm wondering whether the USDA databases may be also good partner in working together so that we can make these connections. So, there are many people actually part of ECBIOData who are also working for USDA. I don't know how to actually create this, how to make this happen. We don't have we have working groups. Maybe that may be a way of getting a closer collaboration together.
Yeah, that's a really great suggestion, recommendation for USDA. We do want to branch out beyond DOE for sure, and we have done that a bit with our collaboration with NEON, the National Ecological Observatory Network, I think is what that acronym stands for, which is under NSF, right? And so again, just we don't necessarily want to create another DOE tool. We want to create a microbiome tool. So yeah, that's a really good suggestion with the USDA.
I'm sure that if you wanted to do that, Tanner, under the auspices of AgBioData, that could be a new working group. We don't necessarily have to have just, some of the ones that we've identified. I think that'd be great. I also wanted to say that I really wanted to thank Montana and her group we stole the idea of champions and ambassadors from you. So thank you for that.
Pleasure.
I think it's a great idea.
A very valuable, framework and structure for outreach. So yeah, I'm glad that's worked out for you too.
Well, we haven't got it going yet.
Okay.
So, Leonard, what kind of topic are you envisioning for this working group? Like metadata standards or collaborations? I think you should
It
add out
sounds like
something, right?
what you're interested in is sort of being able to exploit this technology for USDA databases. So maybe start thinking about like what are, we are not part of the USDA ecosystem, so
Well,
I don't know what
I'm not
sort
trying to exclude the other academicians and companies such as yours. So I think it
maybe
should
you
be
want to
a
take a
bigger
look.
family.
Maybe take a look at that and think about like what needs to get done, in order to be able to, expand to include, databases within the USDA ecosystem.
Yeah.
Are there other, ontologies that need to be integrated? Does it do everything that you hope it would do? How would you, in your USDA sphere, be able to, integrate that information once you have it? And there goes another USDA person raising their hand. So,
Of course.
Well, Tanner, if I can riff on that. So yeah, I was just thinking of a use case there. So I know of people who sequence genomes from eukaryotic species. So like the example I'm thinking about is this group that's sequencing a bunch of bee species. But they're also really interested in the microbiomes associated with those bees for various reasons. But I am not sure if they designed their metadata collection and initial sampling to be able to fully accommodate that. It might, I don't want to misspeak since this is going to be a recording, so apologies if I'm speaking to the people listening to this who might be part of this group. And of course we did this, but I'm not sure that they did. So do you have any cases of people who submit sort of byproducts of sequencing? So essentially when the target organism was a bee or a deer or whatever, but then they also are interested in the microbiome in addition. Does the metadata schema accommodate that? Is that a common use case for this portal?
Yeah, I wouldn't say common because we don't have a lot of host associated data right now, but that is the environment that these host microbiomes end up in is that host associated extension. So we do have one, and I think I feel like it's a goat microbiome, but I could be misremembering. So again, we do have one host associated study on the data portal, and if people want to, expand and be able to share their microbiome data from their host this way, I'm not a host associated person, so the template may not be as well rounded and the tooling may not be, that we may not have as many drop downs as we do for soil. But that's again this like user community feedback that we want to get to figure out like, okay, when did GSE create the host associated extension? How has the research moved forward and that scientific area moved forward, and what has changed, and what can we do to facilitate those community improvements?
Okay, sounds good. I can point them to that user design link that you shared earlier.